Martin mentioned that we have our colleagues from Hawaii joining us. You can see my plot to hold the meeting in Honolulu didn't work out as planned, but there's always the future. So welcome to the meeting today. We put this meeting together to kind of maybe not just show you what the Superfund program's been doing at Berkeley, but to kind of address some critical issues related to PFAS and where we go next. And what I'd like to do today is tell you a little something about how we quantify difficult to measure PFAS chemicals. And, my role within the Superfund Center, my project has nothing to do with PFAS because I like to study oxidative remediation. I like to break down chemicals. I like to turn them into carbon dioxide and water and chloride and things like that. And PFAS chemicals are forever chemicals. And so I spent a long time trying to think about how to do in situ remediation of PFAS and failed. And so we've been studying PAHs and other kinds of important Superfund chemicals like the chlorinated and brominated compounds. And so I got into this question of oxidative. I got signed out of Zoom. How that works.
No, you sure?
Okay, good. Roger's still there. Okay, no, that had a message on the screen. I got into this topic because we had been wanting to get involved. We had been involved in PFAS through research through the CERDEF program for the U.S. Department of Defense, and some of the work that we had been doing in our Superfund project involved with oxidative remediation became the basis for a method of quantifying PFAS known as the TOP assay. And so what I want to do today is I want to tell you about kind of the ways in which we analyze the PFAS for which there are no analytical standards. There's only a handful of PFAS where you can go out and buy an analytical standard and have a quantitative method, and people who work in PFAS know this and know that it's one of the most frustrating things about working with PFAS. One of our students, Dr. Antel, referred to these as dark matter all the time because there's all the PFAS that you could never see. So I want to tell you a little about how we try to see the PFAS that we can't see and how we try to quantify them. And I think the point of this session is to relate this to our efforts to protect public health by turning those measurements into information about the safety of the water. And one of the key issues here is that not all PFAS are equally as toxic. We know that the C8s, the PFAS and PFOA, are particularly toxic, but the other PFAS chemicals, as we're discovering, are still pretty toxic, and the jury's out on them, but it's not like everything is equal in toxicity. And so many of the methods that I'll talk about here today give you information about the total amount of PFAS in a water sample, but that may not be very useful if some of them are like trifluoroacetic acid, which is probably much less toxic than PFOA and PFAS. And so one of the focuses of this meeting is how to turn the kind of information that an analytical chemist can give us about PFAS concentrations into useful information that can determine whether we need to treat a water or stop using it as a water supply or remediate sediment and soil. So that's what I'll try to do here today. This project wouldn't be possible without Lisa and Ned and Fuhar and many of the other people who are in our research group. So we work very closely together. Hopefully, let's see. There it goes. Okay. So PFAS are, this is like a pet peeve of mine, PFAS, and PFOA. I don't know. It's like I can do that. A lot of people can't. It all sounds like PFAS to some people. Okay, so we all know about the carboxylates and the sulfonates. These are the ones that first came to attention. The carboxylates, PFOA is the example of the C8. And the sulfonates, PFAS is the example, but there are a variety of different chain lengths that have been manufactured and that we can find. And most of the time, when we find these compounds in the environment, they occur as a family. That shouldn't be something new to environmental chemists and toxicologists. We dealt with the PCBs, we deal with the PAHs, we deal with the dioxins. You can call them congeners, you can call them isomers, whatever you want to call them. We have to frequently deal with these complex mixtures, both as analytical chemists and as toxicologists. But that's not all there is. I mean, I can find analytical standards for all of these compounds, but there are many other compounds in the PFAS family for which I can't necessarily buy standards. So for example, when we start looking at AFFF, the aqueous film forming foams that are used by firefighters, that's triple alliteration, you can see the sulfonamides in the compounds that are made by 3M. And so the sulfonamides, some of them you can find standards, some you can't. You'll notice that some of them are negatively charged, some of them are going to be positively charged, and some of them are going to be zwitterions. So they have different charge characteristics, and that'll play an important role in the way they partition the solids and the way they move in the environment. But the sulfonamides are not the end of the story. To break the patent that was on 3M's AFFF, a variety of other companies started producing the fluorotelemers. So they put this alkyl group in between and had the same sorts of chemistry at the other end. Again, positively charged, negatively charged, and zwitterionic compounds. Can't buy analytical standards for most of these things. What you see on the screen here is probably the majority of the PFAS that you find in AFFF, the aqueous film-forming foams. There are other compounds, obviously, of the sulfonamides and the fluorotelemers, but they generally have this structure. And what's interesting to me is that when you oxidize them, they mainly get converted to the carboxylates. And so that's why we were interested in this, because we were working for the Department of Defense, and most of the DoD sites are contaminated with AFFF. And that's why we know so much about AFFF, because it was widely released into the environment, and the DoD invested in studying these compounds very early. But that's not the end of the story either. We have things like ethers, for example, like you've heard of Gen X. You see that ether bond there. That compound is also a forever chemical. You're not really going to oxidize it very easily, but those compounds do not get converted to carboxylates and sulfonates when you oxidize them. And we could go on and on with PFAS chemistry, but this gives you an idea of the complexity of these compounds and the challenges in actually measuring them. So what does an analytical chemist who doesn't have analytical standards do? They get creative. And so the first way that people thought about, or one of the first ways that occurred to many chemists to look at measuring the unmeasurable PFAS, was to turn to ways of measuring total organic fluorine. And this was a logical approach because toxicologists and chemists who had been interested in chlorine disinfection byproducts had developed this technology of AOX, adsorbable organic halogens, when they were studying the suite of compounds that are produced when you chlorinate drinking water. Those were chlorinated and brominated. You don't form fluorinated compounds when you chlorinate drinking water, but you do form chlorinated and brominated compounds. And people have been studying this for many decades and finding a whole variety of compounds. And that's been the basis of trying to set drinking water standards for chlorine disinfection byproducts. And so this group, Wagner et al. In 2013, a group out of Germany, took the AOX method, which had been developed for organic absorbable organic chlorides and organic absorbable bromides, and I guess iodides, but they're pretty minor, and adapted it for fluorine. What you do is you take a sample, you extract it, you put it through an oxidation step in an oven at very high temperatures after it's adsorbed on carbon, and then you measure the amount of fluoride produced. And the main way in which this is done is by measuring the fluoride by ion chromatography. That's the AOF-CIC method, which is the method that EPA created a standard method for a few years ago. But you can also measure the fluorinated compounds using PIGY, particle-induced gamma emission spectroscopy. This is a technology that a colleague of ours at Hope College in Michigan developed, Graham Peasley, and he's taken this and popularized it. But there are very few people who do PIGY spectroscopy. Graham's trying to commercialize this and make it easy, but right now there are very few people who can do it. And the advantage of PIGY is that you don't have to actually break everything down to measure the fluorine. And then chemists would say, well, NMR is really sensitive, so we could do CNET F19 NMR and do it, and that is one way you can do it too. The advantage of these methods is that they detect all of the fluorinated compounds, but the disadvantage is they don't tell you anything about the chain length, and as we'll see today, chain length seems to be very important in terms of toxicity. There is usually specialized equipment, so that this oxidation step or the gamma emission spectroscopy or the NMR is not things that most environmental analytical labs have. and the sensitivity is relatively low. Usually you're talking about microgram per liter detection limits here. When we got involved in this back around 2008, when we got interested in this problem, we realized that we could use the approach that we had been using to study chlorine disinfection byproducts. We had been familiar with the method of measuring chlorine disinfection byproduct precursors by chlorinating water and seeing how much THMs and HAAs are produced. Or in our case, we've been looking at nitrosamine disinfection byproducts, and we had developed an NDMA precursor assay. And so we took this experience using hydroxyl radicals and took all of the compounds, the fluorinated compounds in the water, and drove them to the corresponding carboxylates. And that's the basis of the total oxidizable precursor, or TOP assay, which we published in 2012 based upon our efforts to quantify PFAS in runoff from streets in the San Francisco Bay Area. And we found that the ECF precursors, these compounds that were in the 3M patent, made the carboxylates of the corresponding chain length, and the fluorotelomeres, when they were oxidized, the chain length got shortened a little bit in a predictable manner, but they also got converted to the carboxylates. And this method was picked up by commercial labs like Eurofins and Test America and created into standard methods that they offer to clients. So now we're seeing a lot of data on the TOP assay, even though it's not yet become a standard method. And Jean will talk about that in the next talk, EPA's efforts to turn it into a standard method. And I thought I'd also mention another approach that people are using to standardize this, and that's non-target analysis, or sometimes people refer to it as a semi-quantitative high-res mass spec or suspect screening. And in this approach, we use high-res mass spec, and we have various standards, and we know something about the instrument response to those standards, and we convert that into quantification. Even if we don't have analytical standards available, we make some assumptions. And that is a very powerful tool because it allows you to identify unknowns. It doesn't require the standards. The disadvantage of it is you need a PhD chemist who's good at high-res mass spec to do this. And it's semi-quantitative, and we're not exactly sure how to verify the results of it, but it is interesting. So I wanted to show you the results of some collaborative work we've been doing with EPA and a few other labs that have been working on these techniques. We did a round robin recently, and we've submitted this for publication. In this data that I'm going to show you here, we diluted some AFFF and ran it by these different methods in triplicate when we could. And you can see here that at least the suspect screening, the AOF-CIC, and the TOP assay give you higher concentrations of total organofluorine chemicals than the methods where we have standards and quantify with standard methods. And that means that they're able to detect the precursors. I don't know which of these numbers is right, but you can see something about the data that this suspect screening gives higher numbers. The AOF-CIC gives more variability because it's close to the detection limit when we dilute this AFFF down to like tens of micrograms per liter. The NMR method really doesn't successfully detect those precursors, but this gives you a flavor of what some of the data look like. And then we also had a sample of from a contaminated groundwater site, and you'll see here the concentrations are about 2 orders of magnitude lower than our diluted AFFF. So we're talking about a sample that has like 100 nanograms per liter or so of PFAS in it that you can measure with the standard methods, and you can see that the TOP assay and the suspect screening give pretty good results, and they agree with the version of method 1633 that's been modified to include some more compounds for which we have standards. And the AOF-CIC doesn't have any data because it's below the detection limit, and the NMR method is below the detection limit. We've also made some improvements in the TOP assay, and a lot of this came out of Ned's dissertation and another student, Katerina Tsu, who graduated this spring. We've done things like we've added an oxidation surrogate, a C13 label precursor, and we use this to determine that all of the precursors that are present in the sample have been oxidized, and that gives us a lot more reproducibility. And this is something that EPA and others are integrating into their standard methods. And we've also tried to understand better the conditions under which the top assay might fail. It might fail, for example, in a landfill leachate where there's just so much organic matter that the oxidants that you add in the top assay don't convert everything over. But if you have this C13 fossa, you know whether that's the case. And there's simple solutions to these kinds of things, like diluting the sample until you get within its linear range. We've measured the ultra short compounds, the C2 and C3 carboxylates, and that helps us close the mass balance because some of these compounds get converted into shorter ones when we oxidize them. The standard PFOA methods perfluorocarboxylate methods don't detect these ultra short, so it requires often a second chromatography run. And we've also used as a forensic tool the ability to differentiate between branched and linear perfluoro chains in these compounds, and Ned might talk about that a little bit later. And then the last thing I wanted to share about some of the advancements that we've made, and maybe we'll talk about this a little later, is Ned developed as part of his dissertation a mixed mode solid phase extraction top assay, where instead of using just one extraction method, used an extraction method that's capable of separating the positively charged, the negatively charged, and the neutral and zwitterionic compounds from each other. And that gives us insight into the nature of the unmeasurable or the dark matter PFAS that we can't quantify. And what we've learned from that is that, for example, most of the sources of PFAS contamination that have lots of different sources, like things like landfill leachate and sewage, have a mixture of positively charged, negatively charged, and zwitterionic PFAS precursors. When we go in the environment, we find that these positively charged and neutral compounds tend to stick to particles. So they're removed in wastewater treatment plants, they're retained in the soil and the aquifer. The things that are mobile tend to be the negatively charged precursors. So we see these anionic precursors moving in the environment and coming out of sewage treatment plants. So finally, I just wanted to throw up a couple of discussion points, and maybe we'll come back to this in the discussion later, or it seeds some ideas during the day. The first one is that the top assay, which was developed for AFFF contaminated sites, is very well suited for studying those kinds of sites, but it also provides useful information on PFAS in things like municipal wastewater or in landfill leachate. And you'll see that when Ned talks a little bit later about the forensic tools that have been developed. These broad spectrum methods like non-target analysis and AOF-CIC, they're pretty good for hazardous waste, like the waste itself, and samples from the source zone where we expect high concentrations, especially if the source is not AFFF, if it's a specialty chemical from a factory, for example, that the top assay can't pick up. Methods with lower sensitivity, like high-res mass spec, PIGI, 19F NMR, they may be better suited for consumer products or industrial sources. So when you're thinking about like what chemicals are in commerce, these techniques are much broader in terms of their ability to interrogate polymers, for example, and figure out if you have coatings that are PFAS contaminated so you don't have to extract them off. And finally, like when we think about remedial investigations and drinking water surveillance and possibly even the development of drinking water standards, we have to recognize that the analytical methods need to be inexpensive and they need to be widely available in commercial and industrial labs. And so the AOF-CIC and the top assay are currently the best available methods for them. That doesn't mean they're all that's out there. We may end up developing, say, the high-res methods to be useful in this way. But right now, they're out of reach for most people because they're too expensive and they require too much specialized equipment. And I think overall, the methods I told you about today can and should be used in a complementary manner. And so that's my introduction to this topic, and hopefully we build upon it during today.